Hello, welcome to e-learning. I am Dr. Gauri Abhyankar, working as Assistant Professor in Abasaheb Garware College, Pune. Today in this session, we are going to study lysosomes. We will mainly focus our study on structure and functions of lysosomes in plant cells. Lysosomes are membrane-bound organelles and they show lot of diversity in shapes and sizes. The number also is variable in different types of cells. Lysosomes are usually placed near the nucleus. These lysosomes contain large number of hydrolytic enzymes which belong mainly to the class of acid hydrolases. These enzymes are required for breakdown of different types of biomolecules like proteins, lipids, nucleic acids, and carbohydrates. These hydrolases would break down the foreign materials as well as they break down the cellular components also. Now, the learning objectives for this module are: first one, structure of lysosomes; second one, biogenesis or formation of lysosomes; and third one, functions of lysosomes. You can see lysosomes as dark black structures in the figure. Lysosomes are called as suicidal bags because they are able to degrade foreign materials as well as the components of the cell itself. In this picture, you can see lysosomes as red dots. The green bag-like structures are natural killer cells. These cells appear green because they have been tagged by green fluorescent protein markers. Lysosomes are formed from Golgi. Two models have been suggested for biogenesis of lysosomes. According to one model, plasma membrane is internalized first, then it forms endosomes which mature into lysosomes. Other model believes that endosomes and lysosomes are pre-existing structures in the cell. But neither of the models purely can describe formation of lysosomes, and both hypotheses are required for lysosome biogenesis. Lysosomes contain about 40 to 50 different degradative enzymes, most of which belong to the class of acid hydrolases. In the figure, you can see many types of degradative enzymes and examples of each type. These enzymes are active at acidic pH but not at neutral pH. This acidic pH provides protection against uncontrolled digestion of the cytoplasm in the lysosomes. Cytoplasm has pH 7.2. So, if lysosomes are broken, then the acidic pH of lysosome would not affect other organelles in the cytoplasm because the lysosomal enzymes will be inactive at pH 7.2. Now, how this acidic pH is maintained in lysosomes? It is because of a proton pump in lysosomal membrane which actively transports protons from cytosol to lysosomes. The lysosomal proton pump belongs to the family of V-type ATPases. V-ATPases are multi-subunit proteins that assemble as two major ring structures. One ring is a peripheral V1 complex that interacts with ATP, ADP and inorganic phosphates. This V1 complex consists of three parts. First is the globular head piece composed of three alternating copies of subunit A and B. Second part is the central rotational stalk composed of single copies of subunits D and F. Third part is a peripheral stalk made up of subunits C, E, G and H. Second ring structure is an integral membrane VO complex that mediates the transport of protons. The pumping requires energy that is obtained from hydrolysis of ATP. Now, let us see how material is brought in the lysosomes for degradation. The figure shows many different pathways for obtaining materials into lysosomes. Out of these, 3 routes are primarily used by many cells for obtaining materials in lysosomes. These routes are endocytosis, phagocytosis, and autophagy. First, let us discuss about endocytosis. In endocytosis, molecules are taken up from outside the cell. They are received by early endosomes. Some of the ingested molecules from early endosomes are selectively retrieved and others are passed on into the late endosomes for degradation. These late endosomes form mature lysosomes and endocytosed materials are delivered to lysosomes. But this delivery is not a simple process. It involves large molecular machinery with high degree of harmonized events. At the first place, it requires maturation of early endosome to form a multivesicular body or MVB. The maturation events involve several complexes whose activity is highly coordinated in such a way that it will form organelle which can fuse with the lysosome. The major complexes involved in maturation are Corvette complex and Hobbs complex. Membrane proteins are sorted for efficient degradation by the lysosomes, and the sorting is carried out with the help of ESCRT complexes. Finally, membrane fusion occurs between MVB and lysosomes, which is a snare dependent process. Formation of a hybrid organelle and reformation of lysosome both are calcium dependent processes. So this is all about endocytosis. The other degradation process that we will discuss is phagocytosis. It involves degradation of large particles like cell debris or aged cells and microorganisms or foreign particles. These materials are engulfed to form phagosome. Phagosome fuses with lysosome to form phagolysosome, and the materials inside are degraded. One more process of phagocytosis is pinocytosis, or it is also called as cell drinking. This is usually seen in all cells. It is responsible for uptake of solute molecules and smaller nutrients from the surroundings. The vesicles formed by pinocytosis are called as pinocytic vesicles or pinosomes. A very common example of pinocytosis is uptake of water and nutrients by root hair. Now, let us move to third process, that is autophagy. In this process, cells' own parts or organelles are degraded by lysosomes. The process begins with enclosure of organelles by membranes that are derived from endoplasmic reticulum. Such organelles surrounded by membranes form an autophagosome. This autophagosome fuses with lysosome or late endosome, and the material within is degraded. Under starvation condition, large portions of cytosol are non-selectively digested in autophagosome. These digested materials are then used for cell survival when external nutrients are very much limited. The process of autophagy can be divided in 4 broad steps. In first step, membrane becomes crescent shaped and starts surrounding the portion of cytoplasm. Then the membrane completely encloses the portion of cytoplasm, forming an autophagosome. In third step, autophagosome fuses with the lysosome. Finally, due to fusion, autophagosome and its contents are digested by hydrolytic enzymes present in the lysosome. These hydrolases are not synthesized in the lysosomes, but they are synthesized in rough endoplasmic reticulum and are transported to lysosomes through Golgi. See how much is their transport. So this transport is very specific. The secretory vesicles which carry these enzymes do not carry any other proteins or enzymes. The precursors of lysosomal hydrolases are added with a unique marker in form of mannose 6-phosphate group in cis-Golgi network, and they are then transported to trans-Golgi. Trans-Golgi network contains large number of clathrin coated vesicles. These vesicles contain mannose 6-phosphate specific receptors, so they bind to modified lysosomal hydrolases. The vesicles bud off from Golgi and then fuse with late endosomes. The hydrolases dissociate from the receptor and are recycled back to the Golgi. Transport of hydrolases to late endosomes depends on a signal patch present on the enzyme. Mannose 6-phosphate groups are added only to appropriate glycoproteins in Golgi. Two enzymes catalyze addition of mannose 6-phosphate groups to lysosomal hydrolases. First enzyme is phosphotransferase. The enzyme has separate signal recognition site that recognizes the signal patch on the enzyme, and the enzyme binds to recognition site. The hydrolases have N-linked oligosaccharide with terminal mannose. Once the hydrolase binds, then the phosphotransferase adds UDP glucose N-acetyl glucosamine to the catalytic site. The N-acetyl glucosamine phosphate is transferred to mannose and UMP is set free. Then the lysosomal hydrolases with N-acetyl glucosamine phosphate is released from the enzyme. The second enzyme, that is N-acetyl glucosamine phosphoglycosidase, cleaves off N-acetyl glucosamine residue and mannose 6-phosphate is created. Let us sum up what all we have studied. Initially, we started looking at structure of lysosomes and its biogenesis, that is how lysosomes are formed. There are two models for biogenesis of lysosomes. We have discussed both the models in detail. These lysosomes are full with 40 to 50 different hydrolytic enzymes, and these enzymes are active at acidic pH. So how this pH is maintained? So this pH is maintained through a proton pump in the lysosomal membranes. We have seen in detail the structure of the proton pump and how this proton pump works and maintains the pH in the lysosomes. Then for breakdown of materials in the lysosomes, the material has to be received by the lysosomes, or the material should enter the lysosome. There are three different pathways by which the material is received in lysosomes. These are autophagy, endocytosis and phagocytosis. Each pathway we have discussed in detail how it transports the material from different organelles into the lysosomes. Now, along with these three pathways which get the material in the lysosomes, there is transport of these acid hydrolases also, because acid hydrolases are not synthesized in lysosomes, but they are synthesized in endoplasmic reticulum, and from endoplasmic reticulum they are transported to lysosomes through Golgi. Now this transport is very specific and we have seen all steps involved in the transport of these hydrolases from Golgi to lysosomes.